So we're very lucky to have Anthony presenting with us today. Anthony has become a bit of a thought leader in the antibody protein sequencing space. He's given talks and connected with antibody scientists and business leaders around the world for many years. And he's one of the early employees of Rapid Novor and an integral part of the launch of the REpAb monoclonal antibody sequencing service, and now the REpAb Polyclonal Antibody sequencing service. He's an enthusiast in the space, spends his evenings reading and working on antibody science. And during the pandemic, he became a hobby farmer. And so if you also want tips about growing produce, I'm sure he can help you with that too. Without further ado, I'd like to hand things over to Anthony. And we're looking forward to hearing today's talk.
Cool. Thanks so much, Ian. That was a glowing introduction. Much appreciated. So thank you, everyone, for joining. It's great to see you here. I see some familiar names already in the attendees bar, so I'm happy to see you all here. Thanks so much for joining. And to everyone else who's watching this as a recording, hello to you as well, wherever you are and whenever you are listening to this talk. Without further ado, let's get into it. So we're here today to talk about antibody protein sequencing. Our tool of choice in this endeavor has been mass spectrometry. Pardon me, just gonna take a quick sip of water here. So for those of you that are just joining from LinkedIn and haven't really been introduced to us in the past, we've been in the game for quite some time. Rapid Novor's co-founders have been in the antibody and proteomics software game for almost 2 decades. It was about 5-6 years ago that they really decided to specialize in the application of protein sequencing by using mass spectrometry. And in the past several years, we've been able to successfully service well over 300 clients worldwide, many of the major top pharmaceutical companies, and we've successfully completed well over 2,700 monoclonal antibody or protein sequencing projects. Of course, that has been through our flagship REpAb monoclonal protein sequencing platform. And today we're going to talk a little bit more about REpAb, our polyclonal sequencing platform. But for those of you who are curious as well, that's not the only work that we do. We've also begun decoding the immune response by leveraging NovaRig, a novel way for us to kind of do biomarker discovery as well. So in short, what does Rapid Novor get up to? I like to think of it this way, actually, in that we're turning the central dogma of molecular biology on its head. A quick review, it's kind of obvious though, is that you have DNA, which undergoes transcription into RNA and then translation into a protein of interest. And where Rapid Novor fits in nicely is actually by starting at the tail end of this equation and going back. So we can actually derive the full-length amino acid sequence from the protein itself, and then convert that into a DNA, allowing someone to create a recombinant protein of interest and secure indefinite access to that molecule of interest. And this is going to be a recurring theme throughout the presentation, as well as like how we can fit into this kind of equation and plug it into your workflow. So as a quick agenda, we're going to quickly go through a little review of antibody discovery workflows, a comparison between polyclonal antibodies and monoclonal antibodies. I'm going to show you guys the REpAb workflow and examine some case studies and future directions for our team. So as many of you are well aware, there's a lot of different ways you can discover antibodies. I think one of the most common and well-known methods is obviously the hybridoma method, wherein you're going to take an animal of interest, immunize it with an antigen, pull out the B cells, use them with your myeloma, make your hybridoma, culture them, and obtain some monoclonal antibodies of interest. Similarly, many display libraries have been built on the back of phage display, yeast display, and in some cases ribosome display as well. But obviously a very common and well-known antibody discovery technology. And as well, B-cell cloning is becoming very popular as cell sorting techniques and different cloning modalities continue to grow and advance in the field. But of course, I'd be remiss if I didn't mention our workflow, the REpAb workflow. So this is really where the company started to really offer a lot of value in the industry several years ago, was when we started to do this kind of workflow and apply it to protein sequencing, wherein we receive a protein sample of interest, we then digest that monoclonal antibody using a multi-enzyme cocktail. We then take those peptides, run them through our LC-MS workflow, and we actually can derive the peptide sequences using our algorithms, and then reassemble those peptides into full-length antibodies or protein sequences. This has served a lot of teams well with respect to antibody science, because certainly one could discover an antibody against a target of interest, but sometimes that's not the best way. Sometimes people have already done that, and actually they've unfortunately come to a finite amount of that antibody. And in order to successfully recapitulate that antibody's binding activity, they may not be able to access the genomic information. They may not be able to do the DNA sequencing. The hybridoma may have mutated over many years. And in those cases, all they're left with is the actual protein end product. And in those kinds of situations where you've got a really good antibody, you know it's validated, you don't want to go around, sourcing dozens of different antibodies externally, testing them, validating them, and then purchasing them indefinitely. One could sequence an antibody that they've derived in-house and have limited access to using just the protein itself. And that's indeed the premise of a REpAb monoclonal antibody and protein sequencing service. But of course, many of you have come here today to learn about something different, polyclonals. And indeed, we're going to walk through that today. So a little bit of context, again, on antibody discovery. As I'm sure many of you are already aware, polyclonal antibodies have a pretty straightforward antibody discovery process as well, wherein you're injecting the antigen of interest into your animal. You're going to take out a bleed, and you're just going to purify out the antibodies of interest, often using a antigen-specific affinity purification method, and deriving, a very pure and highly specific antibody fraction against your antigen of interest. This has the advantage of being relatively easy to produce, yields some high affinity antibodies, and I think very importantly, has a broad biophysical diversity, and as well as many clones. It's polyclonal. Indeed, there are many individual clones present in that sample. Some of the drawbacks, though, have been with respect to limited supply, some batch-to-batch variation, and they can often be difficult to characterize for obvious reasons. There's a lot of clones in there, a lot of different antibodies, and so the conventional ways in which you characterize antibodies are somewhat limited in that way. In comparison, monoclonal antibody discovery, as I went through earlier, involves injecting the antigen, harvesting the B cells, creating a hybridoma, and performing your clone selection, and then yielding your antibody of interest. This has been taken further through the advent of gene sequencing, and being able to capture the gene that produces the monoclonal antibody of interest, then digitizing said sequence, leading one to be able to express that gene recombinantly in a mammalian cell line of interest. Of course, as I mentioned earlier, REpAb also fits into this paradigm quite nicely by actually working with the end product this time, the protein, then deriving a digital sequence, which can then also be used in order to express the antibody recombinantly and derive a perfect clone. So in this way, REpAb has really filled the gap in this antibody recapture method. So extending on that idea, protein sequencing is offering to fill more gaps. As I've illustrated earlier, we take the monoclonal antibody of interest, we can recapitulate its sequence and binding activity by using a digitized sequence. But similarly, can we maybe apply that same modality within the polyclonal antibody context? And indeed, that's what we're here today to discuss. We're harnessing the advantages of all of the polyclonal antibodies and mitigating the disadvantages therein as well by creating recombinants, by strictly defining the antibody sequences and then creating a cocktail of recombinants that recapitulate the binding activity of a polyclonal antibody. So in summary, we're really trying to merge the best of both worlds. We're taking your poly, which is easy to produce, has many existing products in the market, has a high affinity and broad biophysical diversity, but is limited by batch-to-batch variation, limited supply, issues with scaling, and being difficult to characterize. And we're going back to the monoclonals and taking advantage of the recombinant production that's enabled by securing the digital sequence, and taking advantage of the industry standard processes for upscale production, and mitigating again some of those downfalls. So increasing the high affinity, broad biophysical diversity, and again, enabling recombinant production and standardization of upscale production. So how do we do it? The REPAB workflow works essentially like this, where we take an animal or organism of interest, we extract the serum from that animal of interest, we put it through our previously mentioned proteomics workflow, and in tandem, we extract the PBMCs and put it through an NGS workflow. At the end, we're able to combine the bioinformatics databases that we obtain from both the NGS and the proteomics, and derive full-length heavy and light chain sequences, which we can then actually pair using other proprietary workflows that we've developed in-house, so that we can deliver full-length cognate paired heavy and light chain antibodies, which mimic the polyclonal antibody of interest. So with a little bit more detail, it starts with immunization, in which we're doing an immunization of your animal of interest. We have a preference for rabbits. You can also work with humans as well, but indeed, we get this question quite often about whether or not we can work with other species. I'll address that a little bit later on as well, but just so we can follow along nicely, yeah, indeed, this proposed workflow and the case studies are derived from rabbit experiments, which have gone well. So again, we start with the immunization of the animal. We take a pre-immunization bleed if we can. We do a test bleed, and of course, a final bleed in order to assess titers of antibody response. We then do a protein AG purification for the IgGs, and then afterwards subfractionate a bit further by doing an antigen-specific affinity purification. And of course, we're always testing for titers in these animals using ELISA. And then we bring in screening. This has been a new element to the workflow as well, where we're actually able to screen up to 5 samples during our assessment. And what that means is we take a look at up to 5 different samples, whether they be different rabbits in a cohort, whether they be from the same rabbit, but maybe a different subfractionated IgG pool. That could be from different subdomain purification schemes against the antigen of interest. But in that way, we're able to essentially assess 5 different samples by literally trying to sequence them using our workflow. The purpose of that is really to assess whether or not those samples are going to make it through the full sequencing protocol. Obviously, we're not looking to get in the business of disappointing anyone, so we try to offer a way for clients to screen their projects halfway through the project and without incurring the full cost of the entire project, should the project not be feasible for phase 3. Thus far, it's always been green lights, so we're happy about that. But of course, for those of you who are curious about what might happen during a project, this is a good piece to know, is that you can really send us a number of different samples that we can screen and then process and then determine the feasibility for prior to entering further sequencing efforts, which would encompass deep sequencing, which for us involves putting this, like I said, through NGS and then furthering the proteomics workflow. So in phase 3, we're doing an RNA, bulk RNA seq of the IgGs that are amplified in the PBMC sample prep. We're going to do a combination of the genomics and proteomics databases. We're going to complete de novo sequencing of the most abundant antigen-specific IgG heavy and light chain constructs. And then at the very end, we're going to pair those heavy and light chain constructs into their cognate antibodies. And on the very tail end, we're going to actually make these antibodies. Of course, straightforward process. We're going to do the gene synthesis, transfection into HEC293 cells, expression of those IgGs, and then ship them to you for clone selection. So in short, this is the project overview. This is how we run the project. These are kinds of like the phases and the deliverables and the experiments that are running through each one of these phases. Of course, the question is, how did we do? Well, there's been a number of different applications that have been highlighted to us, particularly in early stage research and development. Of course, people are keen to see this in therapeutic antibody development. The straightforward one is, hey, we could use this to maybe generate some good leads against target of interest. But I find that a lot of interest also comes within the assay development space, and we'll talk about that a little bit more later as well. Further to that, looking to characterize the immune response, doing diagnostic development, and converting a PAB to a MAB or MAB cocktail. And that was kind of the straightforward one that everyone was really keen on, so were we, when we first set out to do a proof of concept study. So we did that. We immunized some rabbits with a peptide of interest and used it as a proof of concept study. And the results are forthcoming. This is some of the coverage that we obtained for that first project, which was completed in the summer of last year. And for those of you that have never seen proteomics sequencing data, no worries. I'll just say that this is really good. This data really shows excellent levels of coverage throughout the antibody sequence that we were obtaining. This bar at the very top in green, orange, and blue represents the variable region, and then the blue is the constant region of our antibody of interest. And below that, you can see the coverage lining up very nicely. In a typical monoclonal antibody sequencing project, we strive to achieve 20 to 30 overlapping peptides over every single amino acid. And in this case, you can see that that threshold was achieved. We were able to really get excellent levels of coverage on this antibody pair, heavy and light chain pair, as well as in this heavy and light chain pair. And luckily, the evidence bore that out. So again, to summarize, we immunized this rabbit with a peptide antigen. We pulled out 3 sequences from this project, so R1 through R3. And as you can see, PD030 is the original polyclonal sera, which was outperformed by these 2 recombinants that we derived, R1 and R3. And we use those antibodies regularly internally, and they are excellent antibodies. They perform well and are fit for purpose and serve us well to this day. So of course, extending that, we've been trying to work with more people, try to derive some more experiments and successes using the REPAD workflow. And we were contacted by a company to do biomarker discovery for their diagnostic project. And so they were looking to use some antibodies in co-immunoprecipitation, where they wanted to lyse some cells, pull out a particular biomarker of interest using an antibody, and then use that as that purified antibody purified mixture as the basis for their diagnostic. And so this obviously is a robust method. It's been made easy to set up, but it strictly requires an antibody that's fit for purpose and can have issues with other kinds of homologous antigens should they be similar to the antigen of interest. And in short, this is the workflow that we're looking at. So taking your sample of interest, pulling out a particular antigen, purifying using that antibody of interest, and in their case, they were using mass spec to quantify the relative abundance of that antigen of interest, and drive a useful medical assay. So this is where I tell you that this project had a crunch, actually. We were putting it through the workflow that I mentioned earlier that had proteomics and genomics, but unfortunately, because it was so time sensitive, we were left with only the proteomics data. Otherwise, the project would have been a no-go, and that was because of sensitive requirements on behalf of that client. So we did what we could. We worked with only the proteomics, and in fact, it added a lot of pressure to the project, but luckily I can say that pressure makes diamonds. So we were able to successfully sequence a number of monoclonal antibodies from that sample, and we successfully validated them using an ELISA, as shown here. So we actually pulled out 6 distinct monoclonal antibody sequences from that sample using only proteomics. And the results were very good, as you can see. The negative control was there. In red, you can see the original polyclonal antibody mix, and the rest of them obviously are performing quite well. The green bar, R5, looks quite nice as well, outperforming that original poly mix. But you know, you're seeing it here for the first time, folks. This is the first time a company has ever, or any team really, has ever completely de novo sequenced several monoclonal antibodies from a polyclonal antibody mixture using only proteomics. Certainly, you know, B-cell approaches and single-cell sequencing approaches have yielded other ways to do this, but I don't think that it's ever been we know that it's never been accomplished using just proteomics, where we're able to sequence the most abundant heavy and light chain pairs and derive and the monoclonal antibody sequences and capture the binding activity in this way. So extending on that idea, we've been in contact with multiple therapeutic antibody development companies and looking to use this, like I said earlier, for antibody lead generation, as well as assay development. And that's really where I think that we fit in. So here's a kind of standard therapeutic antibody development pipeline. You're starting off with, your hybridomas or your mouse B cells or your synthetic libraries, and you're going from many to few. I think that really we fit in nicely within the assay department. So PK/PD assays, targeting specific variants of your antigen of interest, using these or trying to develop surrogate antibodies against your target of interest. I think this is really where we can fit in nicely. And similarly, in sequence optimization, the argument's always there, right? That deriving fully human antibodies from actual convalescent patients could yield a lower development timeline with respect to bringing that antibody from the initial screening through to production. Of course, all antibodies need to be optimized, but certainly we've seen in recent years that the use of fully human antibodies, whether they be transgenic or derived from patients of interest, can shorten those development timelines. And of course, as I mentioned earlier, validated antibodies are well served by our existing REMAB technologies, in addition to the REPAB services as well. Once you've got a really good antibody, you've validated it in your application of interest, and you're just looking to recapitulate that antibody indefinitely. Sequencing its primary amino acid sequence would be useful to that end. And we've got good friends. We've been working quite well with Absolute Antibody over the past several years to develop a nice steady workflow for not only taking that primary antibody sequence, but then further engineering it into a subtype of interest, switching the species or switching the isotype, and ruling out different possibilities with respect to that biological activity. So naturally, once we've seen that it works, the question might be, what do we need to get started? Essentially, we have two categories at this point. We have Go projects, which are, I have a target of interest, I'm looking to do an immunization on some rabbits or I have some patients ready or something like this. Of course, we're happy to talk and work with those projects as well. Or you might have a rabbit or human blood sample which is affinity purified, and you have access to that original blood still, which contains the PBMCs. Like, we can definitely work on those projects right now. And similarly, if you have that blood, but you can also do the NGS, we're happy to work with you where you send us the affinity purified material, and then we can receive the NGS data from you and use that for our genomic database. Or you just have a monoclonal antibody. Of course, we still do that. And REMAB still works quite well. And of course, if you just have a monoclonal antibody of interest, we can definitely work with you and facilitate that project. So following into the Let's Talk category is, I have affinity purified material. I have a PAB. I'm working with a different species of interest, whether that be goat, sheep, llama, mouse, and hamster, or anything else really. In those kinds of situations, I invite you to have a discussion with us about that project of interest. As I mentioned earlier, we were able to sequence several monoclonal antibodies from a polyclonal antibody mixture. We will say that that is N equals 1, and that although it was a great success, and we're super happy about it, and we're happy to share that with everyone, of course, we wouldn't jump to the conclusion that this is going to work repeatedly and robustly for everyone always. We invite you to have a discussion with us, and we'll be transparent about what our capabilities are and what kinds of services we can offer and how we can help you with your development. And similarly, if you're working with a different species than the ones we mentioned earlier, one of the main limitations of our approach with respect to the proteogenomics aspects is that we do NGS, and of course, we're amplifying for the IgG genes in the PBMCs that we work with, and kits that are used for NGS development are somewhat limited. There's kits for rabbits, there's kits for humans. There are not kits, universally available for all species, and so for that reason, we have some limitations in that regard. But of course, we're happy and willing to talk to anyone about developing any of those methods or doing that kind of work as well. So naturally, this extends into our future developments. We're going to do custom NGS development work in-house. We've started that lab. We've hired people to take on that workflow and add those kinds of capabilities in-house so that we can work with other species like sheep, like goat, and that other species that are interesting for polyclonal antibody development. We're going to get greater protein-only sequencing capabilities. Last time I gave this talk, about a month and a half ago, I said that, and luckily this time I give the talk, we actually have fully derived monoclonal antibody sequences from a PAB that were derived using only the protein sequencing capabilities. So those are being developed and are continuing to be developed as they have been since we started the company several years ago. We're delivering more antigen-specific leads. Further to the point, we went from 0 protein-only leads to 6 in that last project, and we actually derived several other sequences using the proteogenomics approach that were independent from the protein-only reads. So in fact, our sequencing capabilities are getting better. Our ability to derive those antigen-specific leads is also getting better. And we're looking at new methods for immunizations. Thus far, we've worked with peptides as well as soluble proteins, but in the future we're happy to consider doing other things like DNA immunizations, VLPs, wholesale immunization protocols and stuff like that. Please, again, we invite you to have a discussion with us so that we can see what we can do for you. So in summary, REMAB is a powerful technology to acquire the amino acid sequence of any pure antibody sample, and by using a combination of REPAB, of proteogenomics, REPAB can provide a library of antibody sequences useful for early stage R&D in therapeutics, diagnostics, and reagent antibody development. Rapid Novor is obviously seeking royalty-free contracts for the use of the technology in the pharma and biotech industries. We're strictly operating on a fee-for-service arrangement. We don't really make this complicated. We try to make it easy for any antibody development teams out there to work with us and get the benefits of the technology in their in-house. So that's it, folks. We'll open this up for Q&A. Again, my name's Anthony. I'm the Director of Business Development here. As you can see in this picture, this is a pre-pandemic picture because I haven't got a haircut in a while. So when we meet on the conference circuit, I might look a little bit different, but I promise I'm still the same jolly fellow. So I invite you to ask your questions, and we'll start with the Q&A.
Thank you so very much, Anthony, for a wonderful presentation. And I'm sure if we were in an in-person audience, we'd have some applause here at this point. But let's do some Q&A. So just to remind you, please type into the chat or into the Q&A box. There are already some questions that have come in. So we'll get to those in just a second. I know some of you are still thinking up your question or phrasing your question. So while we're waiting for those, some more questions to come in, I'm actually going to do a quick poll. So it's a very simple question. And if you could just click on your answer about if you're currently working with polyclonal antibodies, then we'll run through that poll. While that poll is running, maybe we could field the first question, which is how much antigen is needed to purify the PABs prior to mass spectrometry analysis?
Sure. The amount of antigen that we require obviously depends on the project of interest. But I should say that thus far in the projects we ran, we've had to use a couple of MIGs in order to develop that column and purify out the antibodies of interest.
Okay, very good. There's a question about organisms. So rabbits are relatively special in having a major fraction of serum antibodies representing 100 to 1,000 high affinity antibodies. So have you tried on human or other critters?
Yeah, thanks for the question, Dave. Indeed, we have. So we've been working on, well, COVID like everyone else. So we've done a couple of COVID-related projects working with convalescent patients. Those results are forthcoming. And yeah, we're happy to share those results in the future. But like I said, I think that This is why we've changed a little bit of the approach in our screening, because of course we really appreciate samples that are highly convergent. Naturally, we're using mass spec to do the antibody sequencing, and also obviously like the most abundant antibody species are the easiest for us to sequence. So in polyclonal antibodies, which are particularly heterogeneous and like there's no distinct antibodies present that are dominating that antibody population, that's a lot harder for us to work with. But in cases where there is a more convergent antibody response and we're actually able to fractionate that antibody response, we've had good success. So to be more specific, in the SARS-CoV-2 situation, we did work with some convalescent patients that had a very convergent antibody response around the RBD. And the RBD fraction that we obtained was really nice in one particular patient. And we're not the only people that have observed that. Other people who have taken a mass spec-based investigation on this kind of patient population have reported similar results, where some patients have a really nice convergent antibody response, and that makes things a little bit easier for mass spec-based sequencing approaches. And I'll finish that by saying, yeah, we're looking at other animals as well. We've got a couple of projects ongoing with other species like dogs and mice and potentially goats as well. Yep.
Sounds good. Another question here is, how can you discriminate between the different chains in proteomic polyclonal antibody sequencing?
Yep, that's a good question. I figure, I mean, I'm already seeing it now in some of the Q&A, but indeed, people are going to be curious about the pairing methods. So I'll say this: we use top-down approach. We're able to use our mass spec to interrogate these antibodies on the basis of their mass, and similarly, that's what we're doing when we're looking at the chain pairing problem.
Okay. Are there themes in the different monoclonal antibodies to the same target?
Are there themes? I suppose that's a question of homology. Let me know if it's not. But yeah, indeed there are. Like, it really depends though, right? It really depends, I think, on the nature of that antigen. In the projects that we've worked on, which are peptides, like there is a decent amount of homology, I guess, between those antibodies that we're pulling out. Whereas if they were completely independent epitopes, you might see something different with respect to that, those themes or those motifs. Yeah.
Mm-hmm. There's another question which may or may not be related, but it's about can the method tell the number of antibody species from affinity purified polyclonal antibody sample, or be used to evaluate the consistency of affinity purified PABs from batch to batch?
Yeah, that's a great question. One that I would say, to answer the first part of the question, can we tell the number of antibody species from the affinity purified PAB? Probably not. Like, not in a strict sense anyway. What we can definitely do is interrogate unique CDRs and give a sense of the convergence or divergence of that antibody repertoire. But I really wouldn't want us to say something like, we can tell that there's 100 MABs against this target of interest, or like there's this X number of distinct antibodies present. Just because, like I said earlier, with the mass spec based approach, we're really able to derive those sequences accurately because they are overexpressed compared to the polyclonal background. If every single antibody was equally present, it's extremely difficult for us to stitch together any meaningful sequences. And so for that reason, it's difficult for us to like determine the distinct number of antibody species in an affinity purified PAB from only the proteomics. Of course, you can interrogate the B cell repertoire and correlate those, but again, we wouldn't want to say that we can definitely nail down the exact number. In terms of consistency between PABs batch to batch, there might be something there, but we just haven't ever run through that. So I invite you to have a discussion with us afterwards as well, and we'd be happy to kind of explore that possibility with you a bit further.
Okay. Can you elaborate a little bit further on the phase 2, the screening?
Sure, yeah. So in phase 2, let's see, can I rewind? Oh, it's gonna be a minute. Let's go back. Phase 2, we are, sorry, there we go. We're definitely able to, like I said, screen several different samples. So we find this particularly interesting when people are sending us or working with us on a fresh immunization project where they've got several different rabbits against a target of interest, and they want to interrogate like what's the best rabbit, right? Thus far, we've been able to go off of like titers, but depending on what kinds of papers you're reading with respect to like convergence and antibody repertoires, one might argue that titer does not necessarily imply convergence, which is kind of a surrogate for us in terms of sequenceability, for lack of a better word. So in order to mitigate that problem, we've decided to just kind of open it up a bit more and allow people to send us multiple different samples and allow us to evaluate them and determine which sample is best for our purposes of sequencing. And from there, we can put it through the workflow and then derive those folding sequences without having to necessarily run this whole workflow like several times against a cohort of interest. We can screen them all up front and then from there, go through the full sequencing on only those that actually have a good chance of success in phase 3. Yep.
Okay. And can you talk just a little bit more about pulling the most high affinity binders from the sample and how that relates to convergence?
Sure, yeah. So in our, in our workflow, we effectively have two main biases for the antibodies that we're deriving. Just gonna take a quick sip of water. The first bias is that we're only pulling out antigen-specific leads from the proteomics. Because we do the antigen-specific affinity purification, naturally the only antibodies left are those that bind to the antigen of interest. And the second bias is that we're only going to be able to pull out the most abundantly expressed antigen-specific antibodies from that fraction. And so naturally, there's an argument, but there's not necessarily proof around whether or not the body is really like selecting for those antibody species. They're overly expressed, for one reason or another anyway. And we know that they're obviously high affinity binders against your target of interest. So in the workflow, we're going to derive several monoclonal antibodies, and they're going to be high affinity binders. And thus far, the results have shown that they've really served well to recapitulate the binding activity of that original polyclonal antibody. So I think in that way that they're fit for purpose and meeting the needs of recapitulating that original polyclonal.
Thank you very much. There are a couple of other questions, but I think they're similar to some of the other questions, so it seems like they've already been answered. So I won't ask them again. The only other ones are a bit of housekeeping questions about will the material be available after the talk? And so the answer to that one is yes. The recording will be made available on demand after this. So if this is interesting, you can share it with some of your colleagues. So I think we'll wrap up here unless you had any closing thoughts, Anthony. And I'll do a final poll, which is, here we go. Oh, sorry, I put up the wrong poll. I was going to do a final poll about the just feedback on this session, please. So if you could give us some feedback on if you got some value out of the presentation, then that would be appreciated, and it helps us to guide our further talks about this topic. So I'll leave that poll up there for a second and let people respond. While that's running, Anthony, do you have any closing remarks?
Yeah, please have a conversation with us. If you found this information useful, if you can conceive of the use of sequencing a polyclonal antibody, please let us know and keep in touch with us. We're always happy to learn and grow more as a company. And of course, some of that is predicated on what the end users actually want. And we're happy to, try to work this method a bit more so that it might be easier for you to work with us, and so we can fine tune it to meet your end goals. And I guess the last question that I'm seeing from Gustavo here, and I'll answer it quickly, is just how different are the MABs that we identified? Are there usually some variation to the same one, or are they completely different? It really depends on the project of interest. Some have been very similar, some have been quite different. So it just depends on the project of interest. But of course, again, we're happy to discuss further offline if you have any other questions. We invite you to come back and have that conversation with us. So yeah, thanks so much for your time, everyone.
Thank you very much.